Hi, Gretchen. Good morning. Good morning. Good morning, Alex. All right, listen up. So I had several questions on email this weekend about when is your extra credit due, and it's not due today. So I'm actually going to give you Thanksgiving vacation to do it. So it's going to be due on the December 2nd.
That's wonderful.
Okay, so write it down. Extra credit is due on December 2nd. So this morning we're going to start out with a clicker question. So find your clickers. And we're going to answer it silently. Okay, so here's your first clicker question. Okay, it's time to answer the question. I'm going to close the question. All right, now you can chat with your neighbor and talk about your evidence of why the question is right. Okay, I'm going to reopen the question. Okay, it's time to answer the question. Okay, I'm going to close the question. 2 more people should answer. Okay, C is the correct choice. So 93% of you got this correct. The first time around, you were 78% of you had it correct. So any questions about why C is the correct choice? Yes, Jason. I can't hear you. So it says, so you're looking for which one is false. Arrowworms are more close related to cnidarians than to deuterostomes. So you're looking for what's false. Does that make sense? Yeah. You gotta, yeah. False. All right. So today we're going to talk more specifically about the protostomes. So last week we just essentially gave an overview of animals, and now we're talking about the protostomes today, which we introduced last time. So what is a protostome? So we talked about this on Friday. So how would you describe a protostome? Okay, it has to do with the cleavage. So what about the cleavage? What are your choices for cleavage? Spiral or radial? So do they have spiral or radial cleavage? What? Spiral. They have spiral cleavage. Okay, what else do you know about protostomes?
Where they start from.
Where what starts from? The blastopore becomes what? The mouth. So it means mouth first. All right? And then what was the third embryological trait of the protostome? What? Has to do with how the coelom forms. There were 2 ways the coelom can form. Those things on either side of the gut, okay, that were red in the picture, yeah? What do we call that process? No. There you go. Schizocoely. What does that word mean, schizocoely? No? It's in your notes from last time. Okay? So what does it mean? What's a schism? A split. Okay? So the mesoderm forms by splitting. That's what schizocoely means. All right? So the deuterostomes are different. So what did we call it for the deuterostomes? So the notes, it's all in Canvas. So if you guys aren't getting it during class, you need to be looking at Canvas. Okay, so what's it called for the deuterostomes? Someone said it. Enterocoely, and that's an outpocketing of the gut. Enterocoely, all right? So then what else did we learn about protostomes? So we talked about their embryological traits. And then we talked about their nervous system. So where do we find their nervous system? Which is called what? So what kind of nervous system do you have? Dorsal. You have dorsal. So what do protostomes have? Ventral. Okay. So now we know what a protostome is. We just reviewed the suite of embryological traits. So we talked about how they're divided into 2 major groupings, the lophotrochozoans and the ecdysozoans. Okay, so who can remind us, what does this word mean, lophotrochozoan? So zoan just means animal, right? Well, let's talk about the word. What is this lopho part for? John. Lophophore. So what's a lophophore? It's a feeding structure. It's a ciliated feeding structure. Yep. So what's the troch part for? The trochophore larvae. Yeah. Okay. What about ecdysozoans? What does that word mean? Ecdysis. Molting. To molt. So these are the animals that molt. All right. So we also have our protostome, our anterior brain that surrounds the entrance to the digestive tract. Remember I talked about the little song, the circumesophageal ganglion? You laugh, but there's research to show that it helps people learn. And they have a ventral nervous system of paired or fused longitudinal nerve cords. Okay? So in terms of the body, how the body cavity forms, some of the protostomes are acoelomate, some of them are pseudocoelomate, some have a true coelom, and sometimes they have what's called a hemocoel, which is sort of intermediate. Okay? All right, so here's the tree. So we have our two major groupings. We have the Lophotrochozoans. So it shows you here the trochophore, which was subsequently lost in several groups. And then down here we have molting. And we're not learning all these phyla. So we're going to be talking about the arthropods and the nematodes here in the ecdysozoans. And then mollusks, annelids, and the flatworms, or the platyhelminthes. All right, so we're only focusing on a few phyla. All right, so we just talked about how you would decide which lineage. So what would be one of the first things you would look for, Chris? If you knew it was a protostome, how are you going to decide which of those lineages it belongs into? Whether or not it molts. So if it molts, it's an ecdysozoan. And if it doesn't, then it probably is a member of the Lophotrochozoan lineage. All right, so here's your Lophotrochozoans. So platyhelminthes, annelids, and mollusks are the three that we're going to be focusing on. So here is the little trochophore larvae. This is showing you what a lophophore looks like. So on our field trip on Saturday, we probably saw some colonial bryozoans. They were like the very small things that were encrusting, and that you can't really see them without a microscope, a dissecting scope. Okay, so but this is one that's larger. So they have these U-shaped feeding structures. All right. Okay. So these are usually organisms that don't move. That's what this word means, sessile or sessile. These are not, they don't move. Sedentary, they stick to something. Here's another picture of, this is another phyla called a phoronid that also has a lophophore. Okay? So it's a ciliated feeding structure. Whoops, that's not what I wanted to do. So the evidence that we have currently shows that the lophophore appears to have evolved independently several times. That's one possibility. Or it's ancestral and has been lost by some groups. So which one of those two ideas fits the parsimony principle? What? That it's ancestral. Yep. Okay, so if it evolved once and then was lost, that would be a simpler explanation than if it evolved independently multiple times. All right, so most of these things don't move as adults. Some of them have a larval stage. You can find these, I've seen these up in the San Juans, the brachiopods, which are also called the lamp shells. And there's a few places to find these guys as well. And these are very common, the bryozoans. All right, so, and in lab we looked at the specifics of the, do I come back to that? I think I do. We're not going to, yeah. In lab, we looked specifically at the body plans of your annelids and your mollusks. So today we're kind of doing the overview. So here we can see all of these ecdysozoan lineages, and they all molt. And we're going to be talking about the arthropods, which have jointed appendages, and then the nematodes, which are also called roundworms. Okay, so here's your flatworms, platyhelminthes. These are the Lophotrochozoans. So here's another picture of a bryozoan. So we're going to talk a little bit about the flatworms because we didn't talk about them in the lab. So they have no respiratory system. They have no way to transport gas around. So we said they're flat, and that helps them get away without having many organ systems. Each cell must be near the surface. All right? So they're dorsoventrally flattened. Okay? They have an incomplete digestive system. So that means there's only one opening. So what goes in comes out the same opening. And that's fairly common in the invertebrates. And here's a picture. So if you ever dissected the planaria in high school, anyone did that? No? It used to be common in high school lab. The planaria you can cut in half and they will regenerate. Truly. Some of them are parasitic. So we have some that are free living, that have heads. And most of those, we call them turbularians. A lot of them are parasites, so tapeworms and flukes. And they actually absorb digestive food from their host, so some of them have lost their digestive organs. And some of these are disease-causing parasites with complex life histories with multiple hosts, similar to what we saw with the Plasmodium. And here's one example from the book. This is a tapeworm. Well, okay. Don't take everything quite so literally, Serena. Let's say the bear was there the day before the fisherman. It's just showing you how it moves. Oh my God. All right. So here we have the copepod. So we have actually 3 hosts. Actually, we have the copepod and the fish and the mammal. So we saw with the Plasmodium, we saw 2 hosts here. This is even more complicated in that it has, we have another fish. We have 4 hosts. So we have a very complex life cycle here. Okay? So we're not learning the details of the life cycle. But you someday you may take a parasitology class and you can learn all the gory details of these amazing things. All right? So any questions about this? All right. So ecdysozoans, they have, when there's a cuticle that's thickened with proteins and chitins, we use this word exoskeleton to refer to it. And here we have some examples of molting. So they literally crawl out of their exoskeleton. And then, so this is the famous soft shell crab. It's a crab that just crawled out of its molt, and it hasn't hardened its new shell yet. So basically, they build their new exoskeleton. They build it inside, and it's all kind of folded up, and it's not hard. They crawl out of the old one, and then there's a biochemical process that occurs that makes they take on water and unfold it basically, You know, think of accordion pleats, and then you unpleat it. And then it's hard. And then after some period of time, they get a molt again. So you can go to the beach and find crab molts pretty regularly. Yes? What kind of insect would you call a cicada? What kind of organism would you call a cicada? It's an insect. So yes, it's an yes, sorry. That didn't come out right. I need more coffee. All right. So, protostomes are very abundant. All right? And you want to go back through your lab things and make sure you understand the basics of the body morphology of the ones you looked at in lab. So, we eat them. Right? You like shellfish, bivalves, shrimp. Copepods and krill. So we don't eat these directly, but those are a very important part of the food chain for other organisms that we do eat. Ecologically, these types of organisms are filling all of the niches: consumers, predators, scavengers. Obviously, they're not producers. In terrestrial systems, we have insects. They're very important pollinators of crops. Most of the control of insects comes from other insects. Insects and spiders eat insects. So if you spray pesticide, it kills all the bugs. It kills the bugs that were controlling the bugs that were eating your crop. All right? So it's not a good idea in the long term. So what are earthworms doing? What do earthworms do? What? They're detritivores. So what ecosystem service do they provide? Decomposition, yeah. What else, though? The earthworms don't do that. Bacteria do that. But they are helping enrich the soil, right? They're breaking things down, and they are aerating the soil as well. Okay? All right, so this is a graph. This is from a different textbook because it's just really visual, and it's red. It's Wake You Up this morning. And everything in red and purple, everything that's red and purple is a protostome. Okay? And a lot of those are insects. Well, they're breaking this out. So here we have nematodes, mollusks, platyhelminthes, annelids, and ectoprocs. So the only things that are not protostomes are gray. So chordata, that's us, echinoderms, and then the cnidaria and the porifera, and then other phyla. So protostomes are pretty important. That's the takeaway message from the graph, right? All right, so we want to talk a little bit about how organisms move and how this is associated with their trophic ecology. So in the early Cambrian, we had fast-moving prey and predators, and then we saw the development of skeletons. Skeletons help animals move more rapidly. The earliest skeletons were hydrostatic skeletons, so fluid-filled cavities. And if you, a lot of organisms still use this. So earthworms have a hydrostatic skeleton. Anemones have hydrostatic skeleton, and they suck in and they go out, and they're have a lot of range of mobility based on that hydrostatic skeleton. Well, that's still very common. So you can think of it sort of as like a hydraulic fluid, right? It's under pressure, helps things move. And they have more control depending on the types of muscle layers they have. So earthworms, for example, have two different types of muscle layers. They have longitudinal and circular, all right? And so they can crawl. And they do that by alternating. You know, they'll contract and relax, contract, relax, the circular versus the longitudinal muscles. Other things like the nematodes. If you're looking at something in the lab with a microscope, how do you know if you have found a nematode? Anyone ever seen nematodes? You didn't see nematodes. They're pretty small. Sometimes you might have seen them in lab. They're called little vinegar eels. They're not eels. So they can only thrash. So if you see a little worm and it's just thrashing back and forth, that's a nematode. It only has longitudinal muscles. So it doesn't have the antagonistic ability of the circular and the longitudinal that the earthworm has. Okay? So the types of muscles they have and how they can use them are going to help control their hydrostatic skeleton. We had ciliates in lab. I didn't see any. They would have looked like little worms that were just thrashing. I didn't see any in lab last week. All right. So you can also think of the shells of a bivalve as an external skeleton. Or the gastropods, the snails, have one shell. And then we have endoskeletons. So vertebrates have endoskeletons, and echinoderms have endoskeletons. Okay? So that means it's inside instead of outside. And then some organisms have these very extensive exoskeletons. So the arthropods have the most sophisticated in that their limbs are also jointed. All right, and then we see segmentation in multiple animal groups. And segmentation is important for movement, for burrowing, for swimming. Humans also have segmentation. All right. So in lab, you should have seen that the segments were the same. At the front, some of them had organs, but you should have seen 5 pairs of hearts in your worm for a total of 10 hearts. What animal did you look at in lab last week where the segments were not the same? The crawfish. So they're more specialized. And insects have the same thing, where they have different regions and the segments don't look the same. All right, so another eye clicker question. Oh. I think it's based on, this is random. Okay, I'm going to come back to this. Let's go for this question. The order of my slides is. It's hiding under this blue thing. There we go. Thank you, Gretchen. Okay, it's time to answer the question. All right, I'm going to close the question. Okay, now you can chat with your neighbor. Okay, I'm going to reopen the question. Okay, should answer the question. All right, so the right choice here is D. Okay, so segmentation in annelids and arthropods is an example of homoplasy. So that means it evolved separately, independently. Okay, so this is important. This is one of the big changes that happened when we started looking at this molecular evidence, and they came up with these new clades, the Lophotrochozoans and the Ecdysozoans. So find your worksheet from last time, which was 86, I think, page. This, page 86. All right. So this is so we're talking about the very last question, question number 15, which asks you about differences between the old phylogenetic tree, which is what you saw in that animation that you did, and the new phylogenetic tree, which is in your book. Okay. So here you see in the book, if segmentation in we have arthropods here and we have annelids here, okay? And so for a long time before the molecular data was used, the annelids, the arthropods, and the mollusks were grouped together, okay? And they were grouped this way. They were called sister phyla. And they were grouped this way because these two share what feature? The annelids and the mollusks share what? Yeah, not that though, because lots of things have coeloms. So what made these? Why did they put these two together? They're still together, so they're together. They both have what? The larva. They both have the trochophore larva. So these two were together for the trochophore larva. Okay? And then, I want to draw it like this. Okay, and then these two were together, why? Segmentation. Okay? These two were together because they're both segmented. So when this new molecular data started to be published, showing this split, the ecdysozoan and the Lophotrochozoan, a lot of people that had been invertebrate zoologists for 20 or 30 years didn't like it. What are those new kids in the lab with all that molecular stuff? What do they know? Seriously, I'm not kidding. Okay? And so this was pretty controversial for quite a while. And so these studies, so what people did was they started looking specifically at the genes, determining segmentation in the annelids versus the arthropods. And so it's now been shown that segmentation did arise independently in those groups. Okay? And humans also, we also have some segmentation as well, and again, independently. So segmentation arose at least 3 different times. Okay? And so the parsimony principle is not always accurate. And so the new trees are now widely accepted, but they weren't for initially when the data first started to be published. Okay? So that's one of the biggest differences between the old tree and the new tree. There's a few other ones as well, and it is more to do with the way they arrange the acoelomate organisms as more primitive than the pseudocoelomate versus the coelomate. And in some cases, the acoelomate condition and the pseudocoelomate condition are secondary. So they had a coelom at one point in time, and then they lost it. They reverted to the ancestral state. Okay? Does that make sense to people? So did you write that down on your... All right. Okay. So let's go back to... Oh, this is where I want to... All right, so let's talk about this as well. So this is another interesting thing that we've learned in the last few years as we've been able to look more and more at the molecular data, is that the origin of insect wings, the development of the wing is governed by the expression of the same gene as the appendages of the crustaceans. Okay, so remember these are major groupings in the arthropod phylum. The crayfish are in Crustacea, which is sometimes considered a subphylum, and the insects are in a different subphylum called Uniramia, which is mostly insects and has a few other things like millipedes and centipedes in it. So we now have molecular evidence to show that this part of this leg that forms from this PDM gene product is also, so you can see it turns blue, that shows you where the gene's being expressed. It's also expressed in the wing of the insect. That is cool. So, and that's fairly new information as well. Because, so why do we care so much about the insect wing? What's significant about the insect wing from an evolutionary perspective? It can fly with it. What?
Also, how does it develop? How do you get something that's a full wing on something that's...
Okay, so we're not going to, we don't have the, no, we're not going to go there. You can go look up the research papers and learn all about it. But what's significant about the fact that the insects fly? What did flight allow the insects to do? Well, they do pollinate plants. What'd you say? Fill a different niche. So one of the reasons they're so successful is because insects live everywhere. And the flight opened up this whole new set of niches and resources that they could then utilize. So it's an evolutionary innovation that helped them to expand into a variety of niches and speciate. Okay? So you, and one of the things that they do, obviously, is they do pollinate plants. And they use the plants as a source of food, the nectar, and so if they weren't able to fly from plant to plant, that wouldn't work. There's other insects that fly that are predatory. Okay? So it opened up a lot of new niches when they evolved that ability to fly. All right. So some sort of general trends in protostome evolution. So we've seen, we saw this with the tapeworm, that they have this very complex life cycle. Metamorphosis, insects metamorphose. We're not going to worry about the difference between the different types of metamorphosis, but you should know, obviously, that Crustacea insects metamorphose. We saw the complex life cycle with the tapeworms. And that, again, allows the different stages of the organism to occupy different niches. So they're feeding on different food at different stages. So there's less direct competition with individuals of the species. You can think of this as resource partitioning. And this is going to reduce their intraspecific competition. They made this aquatic to terrestrial transition repeatedly. So we see metamorphosis in both marine and terrestrial forms. We have lots of different types of these organisms that are parasites, and that again allows them to exploit additional niches. Lots of different types of feeding structures in the protostomes. Predation favored the development of hard external body coverings, also contributed to new methods of locomotion, so the jointed appendages of the arthropods help make them very successful. What's a disadvantage to having an exoskeleton? And this was one of your lab questions. What's a disadvantage? Nick?
Armor is heavy and it requires more energy to move it around and things like that. So when you use it that way, it takes up from what you...
Could potentially, yeah, energetic. There's an energetic cost. Yes. Good. When they're molting, they're very vulnerable to predation. So it's a good protection when they're not when their exoskeleton's hard, but there's a period of time where they have to crawl out. Basically, they crawl and hide until the new shell's hard again. Yes.
Can they repair it, or
Well, when they molt, they make a new one. Can they repair it? Not that I know of. I don't think so, but I don't know of that for sure. Right. Okay, other questions? All right, and so we see better locomotion, so we have segmentation in the protostomes, helping them be able to move. All right, so here's another clicker question for you. So any other questions in general about the protostomes? So you want to understand that the embryological development and that colored slide in the slides from Friday is the best way to do that. And then within the protostomes, you need to know your two clades, the Lophotrochozoans and the Ecdysozoans, and the trochophore larvae versus the molting, and then the details of the organisms that we looked at in lab plus your flatworm. Okay? All right. So here's another question for you. Oh, but I lost my thing. This little toolbar likes to go away sometimes. All right, so there you go. Which of the following statements is false? Did people already answer this? I'm going to... All right, there you go. Pick E all you want. You're not getting any points for picking E. I can't see anything up here. Okay, time to answer the question. Okay, I'm closing the question. All right, do a quick think, pair, share with your neighbor. Okay, I'm going to reopen the question. Remember that you're looking for what is false. You're looking for what is false. So which one of these is not true? Okay, D is the correct choice. All right, here you go. Here's another one, which is false. You're looking for what's not true. Okay, time to answer the question. I'm going to close the question. Okay, go ahead and chat with your neighbor. Okay, I'm going to reopen the question. Okay, let's answer the question. I think you're doing that to yourself, Alex. All right, I'm going to close the question. All right, what's the right choice here? A. What's a thorax? So that was something you should have seen in lab, is where the wings are attached to your insect. So what's your evidence for choosing A? What's your evidence for choosing A? What information did you use? The other ones were true? What about this figure? Would they all have this gene showing up if wings had evolved independently in different groups? No. Okay? All right. So I'm going to pass back the exam. I think most of you probably saw your grades in Angel.